On May 1st of last year, a lab in the United States confirmed the first case of the citrus greening disease in the Central American region. Belize is one of a few countries around the world that has for some time now been carrying out consistent health monitoring of its citrus industry to detect any disease that could affect the quality of its citrus for the local and foreign markets. But in spite of our best preventive efforts, one of the first, most destructive and costly plant diseases of this new millennium, thought at first to have been isolated in Asia, is now in our backyard. Citrus greening, also known as Hunglongbing, HLB, or yellow dragon disease, is one of the most serious citrus diseases around the world. It is a bacterial disease that is identified in citrus plants. It has no threat to humans or animals, but it significantly reduces the production of the citrus plant, and it can even kill the tree. Once confirmation has determined the presence of the phloem-limited bacteria on the plant, there is no cure. Officials at Belize Agricultural Health Authority see this as an extremely dreaded disease that is high on their chart of plant diseases.
This is extremely high on the chart. We've had our, I shouldn't say fair share, but we've had our share of the uglies, if you may, in the past, in the recent past and way before. It's no surprise agricultural systems get affected, new introductions, etc. But this is one, perhaps, at least for citrus, this has been one of the most dreaded ones.
Although there is no cure at this time for the disease, steps are being taken to lower the threat to this important commercial industry. Towards this end, funds have already been identified and accessed. With leadership from the Ministry of Agriculture, funds were immediately obtained through the International Regional Organization for Plant and Animal Health, or IRSA, to develop and carry out a program to mitigate the impact of the disease. Over $1 million, for example, is currently being utilized through emergency declaration assistance from neighboring IRSA, Mexico. Henry Anderson, CEO for the Citrus Research and Education Institute, explains how the funds are being used.
It's going to be used for education, to let the growers understand what's happening. It's going to be used for surveillance, to continuously monitor the spread of disease. Since you can't cure it and you're trying to control the spread, you need to always be going out there to see how it's moving. Okay? It's also going to be used to take out backyard trees. Now, the psyllid doesn't know if it's a commercial grove or a backyard. So where you have backyard, meaning in your homes, you have trees that are in our yards, trees that are positive or have psyllids. There's a campaign that's going to be launched to talk about Belizeans who have these trees, to say, listen, let's take this out so we could secure the threat or prevent the threat or lower the threat to our commercial industry. It's also going to be used to assist the CJ and CRI to develop a new nursery enclosed, all right, because Baja has mandated, as the rest of the world, that new nursery plants need to be grown under cover. And the reason for this is that the bacteria is very hard to see in young plants because the titer of the bacteria is very low. Okay, so the only way you could ensure that a plant that you're putting in the field doesn't have the bacteria is growing it on the screen and taking control members. So some of these funds will be used for that.
Immediately after funds were secured, a limited baseline study was conducted to determine where the infected trees were most prevalent in Belize. Mr. Lemuel Palacio is the Citrus Greening Control Program Coordinator. He talks about the findings of that initial study.
That survey established that the main inoculum of HLB in Belize is in the small communities and backyards along the coast. So since the beginning of this HLB program, we have given priority to backyards and small farms, because based on that delimiting survey, there is where the main concentration of the infection lies.
With those initial findings, a comprehensive strategy was established to achieve the project's objectives. Those include monitoring and surveillance for HLB, removal of inoculums, HLB-infected trees in backyards to reduce source of infection, control of vector populations to reduce psyllids, production of certified planting material for new groves. Since November of last year, the project has implemented an approach designed to manage the infection among existing plants, while preventative steps are put in place to minimize infection among future plants. A team of highly trained staffers go out and into the field to examine, identify, and collect suspected samples for testing. The team of field staffers responsible for monitoring the southern zone is headed by a technician provided by Citrus Growers Association. On any typical day, they venture into citrus orchards and community and town backyards to do their inspections. As the team journeys into a suspected backyard or field, they can identify suspected trees quite easily. Samples are carefully documented and properly packaged before moving on to another location. Ms. Veronica Manzanero Mahil, Coordinator, Belize Citrus Improvement Program, spoke on the symptoms seen in citrus plants affected by this disease caused by the aggressive bacteria.
The typical symptoms of the disease is blotchy mottle, which is basically a blend of different colors, a light green with a dark green, and that is very typical. Also, it causes very small and deformed fruits in some instances, and we have a very acid taste to the fruit. And essentially that's it. I mean, the complexity about the disease is that it can easily be confused with micronutrient deficiency. So people who are scouting for the disease in the field must be highly trained, and it takes at least maybe about 3 months for somebody to be very proficient in recognizing field symptoms of the disease.
At least 8 leaves are collected from suspected trees, stapled together, and placed in a plastic bag. Each sample is labeled with an information sheet which includes the name of the scout who collected the sample, GPS reading, height, age, and variety